My name is Dr. Bob Van Son from Penn State University, Extension Veterinarian, and I'll be the moderator today for what should be a very interesting and very important subject relative to violative drug residues in our dairy herds. So let's go ahead and get started right away. We got a lot to cover today. We'll start in with Dr. Craig Schultz. I'll introduce Dr. Schultz. Dr. Schultz is a native of northeastern Pennsylvania, grew up on a 60-cow registered Holstein family farm. He attended Cornell University, did his bachelor's degree there, and then went on and completed his doctorate of veterinary medicine. And then he went into private practice in the northeastern section of Pennsylvania. And following his practice experience, he became a veterinary medical officer with USDA FSIS, which is the Food Safety and Inspection Service. And he worked at a large broiler slaughter facility in meat inspection. From there, he took his expertise as a veterinary medical officer to a large slaughterhouse facility in northeastern Pennsylvania in Wyalusing, and put together a really nice program of reducing residues and blemishes for that facility. And with that expertise, Pennsylvania recognized that, and now Dr. Schultz is acting as the director for the Pennsylvania Department of Agriculture with the Bureau of Animal Health and Diagnostic Services, and acting as our state veterinarian. So with that, Craig, go ahead and start your presentation.
Thank you, Bob. I'm going to spend just about 10 minutes at the beginning of my program talking to you about some of the basics of residue avoidance and from the perspective of someone who's worked in a slaughter plant and has an appreciation for what comes in there. And then I'm going to spend the last 10 minutes describing the slaughter plant activities and how surveillance occurs, how residue surveillance occurs in the slaughter plant. The performance bar for residue avoidance has been continuing to rise, and in recent years we've seen some real changes in the way that residue surveillance occurs. On the tissue side, in 2005 there was a pilot project done for anti-inflammatory drugs, including flunixin. The results of that pilot project, it involved about 1,000 cows, was nationwide, done by FSIS, the Food Safety and Inspection Service. And they looked at 710 cows that they considered to be suspect for flunixin residues. That was cows that had musculoskeletal disorders, cows that were lame, cows that had certain inflammatory conditions associated with flunixin use as well. And out of that 710 that they surveyed, they found 50 that had violative residues in muscle and or liver tissues. So that was a concern. In addition, they looked at some 251 healthy cows that would, as a control, that were not in any way associated with the risk factors or the antemortem or postmortem presentation that would be associated with the flunixin residue. And when they looked at this group, they found about a 0.8% incidence of violative flunixin residues in these perfectly normal cows that would not have received surveillance. So that was a major concern. If you consider that we have a possible 0.8 surveillance, 0.8% incidence of flunixins across the population, and we're killing about 2.5 million dairy cows a year, that would be some 20,000 residues a year just for flunixin. And right now, nationally, the number of violative residues that are observed or identified by FSIS is somewhere in the neighborhood of 1,500 to 2,000. So just for an idea of what could be out there and why we have concerns about particular drugs. In addition to that, we've got some concern about milk, and FDA in late 2010 became concerned about possible drug residues in milk that were not being picked up by their routine surveillance. FDA's surveillance is pretty much limited, or has been in the past, pretty much limited to beta-lactam drugs. They developed a procedure, a rapid, highly sensitive procedure for some 26 animal drugs that not only included antibiotics, but also included antiparasitic drugs, antihistamine drugs, and anti-inflammatories like Penicillin. So that program was supposed to be driven by cattle or dairymen that had a history of violative tissue drug residues in their slaughter cattle, and they were going to target these particular dairies and take a look at the bulk milk in these dairies, take samples of the bulk tank to see if they could find similar residues in the milk. So this is another major concern that's giving us all cause to take a much closer look at our residue avoidance strategies and how to best address the potential problem of violative residues both in milk and in beef. A little bit about surveillance. The dairy cow is a surveillance target, and this information that you see here on the slide is pretty much what drove the FDA toward their interest in milk. 7.7% of the cattle slaughtered in the United States are dairy cows, but they account for 67% of the total violative residues detected by FSIS in all slaughter cattle. So we certainly have some risk associated with the dairy cow. And why do we have that risk? Well, one, the age at which dairy cows are slaughtered. Many of them are in advanced age. The way we use them, the multiple lactations and parturitions that cause them to be subject to a number of inflammatory conditions, mastitis, metritis, metabolic conditions like abomasal disease. All of these are potentially associated with animal drugs and animal drug use and the potential for violative residues. And certainly the individual value of dairy cows is an important factor, because these animals are quite valuable, and we're therefore going to be willing to invest a considerable amount in keeping these cows healthy and well. So these factors increase the complexity of on-farm residue avoidance strategies and how we deal with residues on the farm. Residue avoidance in dairy cows is much more complicated than it is in other species. The conventional wisdom with dairy has been simply looking at the drug that was used, reading the label, and determining its pre-slaughter withhold, and assuming that everything would be okay if that pre-slaughter withhold period was met. That's a bit of an oversimplification in today's environment. In 1994, the Food and Drug Administration passed an amendment or an addendum to the Food, Drug, and Cosmetic Act called the Animal Medicinal Drug Use Clarification Act, which fortunately gave veterinarians the opportunity to use drugs on an extra label basis, but with that also came a number of requirements for the veterinarian in the way these drugs were used. Required a careful diagnosis. It required them to determine that no other labeled drug was available for that particular use that would justify the extra label use. Gave them some responsibilities in terms of maintaining the identity of treated animals, and also charged them with developing and working with producers to determine an extended scientifically supported pre-slaughter withhold. So bottom line, as we're dealing with more complex disease situations in our adult dairy cattle, we've got a much greater challenge in avoidance. Some other factors that make it difficult and sort of fall under the definition of the veterinarian client-patient relationship and how that is to function in using drugs on an extra label or prescription basis. We have to take into consideration the dosage and whether or not there has been overdosage accidentally in some cases, but if it occurs, that is extra label use, and we have to have the veterinarian client-patient relationship functioning to deal with the pre-slaughter withhold. Same way with duration of dosage. If the label says 3 days and you use it for 5 days, we've got a concern there. Route of administration, if the label says injected at the base of the ear and it's been injected intramuscularly in the neck, similar VCPR, veterinarian client-patient relationship, is required. Use of multiple drugs over time in treating chronic conditions. Another area where we can get into trouble with determining an affected slaughter withhold. Certainly cattle that are coming to slaughter in a distressed condition, and this is a little heifer that came to Wyalusing several years ago that had Johne's disease. This is something that we see on a regular basis in the slaughter facility, and certainly this animal is compromised. If she had a mastitis problem or some other inflammatory problem in the last few weeks and she was treated with an antimicrobial or an anti-inflammatory, because of her compromised condition, she may not metabolize that drug as fast as she should or as adequately as she should, and there again you need the aid and assistance of your veterinarian in deciding what would be an appropriate pre-slaughter withhold under those circumstances. Some critical factors in residue avoidance. If you talk to FDA investigators where they have done follow-up investigations on violative residues, on-farm investigations, they will tell you without exception that the most common problem that they identify is a lack of adequate record keeping on the farm. And it's extremely important that in your record keeping, in addition to having all the accurate records associated with the animal, that you have established a documented veterinarian client-patient relationship, that there is an involvement of a veterinarian when extra label drug use or when prescription drug use occurred, that you have documents, including perhaps some treatment protocols from your veterinarian in your file to support that VCPR. Over-the-counter drugs, you've got to follow those label directions exactly as we described before. If you vary from those directions, you have a potential problem without a VCPR. So now let's go to the slaughter plant and just talk a little bit about how surveillance is done at slaughter. Not all dairy cows that go to slaughter are screened for residue, for violative residues. I think that's a misunderstanding by many who ship cattle. Only cattle that have at-risk antemortem and postmortem conditions are going to fall into this category. And over here on the left, we've got your typical distressed cow coming into the slaughter, dull, dehydrated, sunken eyes, abnormal posture, probably has abomasal disease or some other inflammatory condition, mastitis, metritis. In general, not well and probably acutely ill. This animal is going to be suspected and receive an individual veterinarian-performed disposition and receive a residue screen on the postmortem floor. And then over here, we have cattle that go through that antemortem process with absolutely no abnormal findings. And once they get to the postmortem floor, we find a big jugular furrow injection site here. Didn't show up on antemortem examination, but once you get to the postmortem floor, these kinds of lesions are identified, and we're going to treat this as a potential for residue screening. So this animal will receive a screening test for residue. All plants in the United States that slaughter cattle must have a hazard analysis and critical control point program. Under that program, the establishment has to identify hazards that occur in its slaughter process. And one of those hazards can be violative residues. Certainly in market dairy cow plants and Bob veal cow plants, violative residues are usually in the hazard analysis and critical control point program. The goal with defining a hazard is to either establish a critical control point or a prerequisite program that addresses that hazard, and then within that you establish limits, which if those limits are exceeded, you take corrective actions to keep that hazard under control and potentially reduce it to an undetectable level. Under HACCP, the industry is responsible, and most industries most firms that have a HACCP program that addresses residues have in it some mechanism for addressing those violations, reducing those risks, and ensuring the safety of their product. And what they're going to do is to trace the violation back to the producer, to provide that information to the regulatory agencies, and then also contact that producer to get some corrective actions and preventive measures so that the violation will not recur. Industry is also required to avoid high-risk sources of animals. So if an industry is receiving multiple animals over a long period of time from the same source, they are expected by the inspection process with the documentation in their HACCP plan to take appropriate measures so that they avoid those sources of animals. This is just sort of a graphic presentation of how we can see the value of HACCP as opposed to the traditional means of investigating residues. That the HACCP plan has allowed this feedback loop to occur, the slaughter plant identifying violations and getting back to the producer. And once that producer receives that feedback, they're going to take the appropriate avoidance measures to make sure that doesn't happen again. And if you look at the black line under the traditional type of FDA investigation and where we didn't have HACCP in place, sometimes that FDA investigation took 6 months to a year to get back to the producer, and by that time most of the information was forgotten, and the feedback that was received wasn't nearly as meaningful as it was through the HACCP process with direct contact of the producer. A few tissue residue points to remember. One is that only high-risk animals are tested, as I said before. And how many of those are there? Well, if you're looking at high-quality dairy cows, tail enders, sterility cases, cows that are milked off and coming to slaughter pretty healthy, or market beef cows, beef breed cows that are not handled as intensively as dairy cows, you're going to see an incidence of at-risk conditions, both antemortem and postmortem, somewhere around 5 to 8%. With lower quality lean dairy cows, a lot of transition cows that you typically see in some of our large dairy cow plants, you can get up to a 15% incidence of those at-risk antemortem and postmortem conditions. Tissue samples from screen-positive animals are submitted to a federal laboratory for analysis. We do screening in the test in the plants. That screen only tells us that there's the potential for a violative residue to exist, but to actually determine what that residue was and the amount that was there requires qualitative and quantitative analysis at a federal laboratory. Interestingly, if you look at all the screen-positive animals that go in, and I'm basing this on my experience from the plant I was in northeastern Pennsylvania, but about 20% of the animals come back from the federal laboratory that are negative, that we don't find any residues on at all. Another 60% come back that are positive, but are below tolerance and therefore are passed. And then another 20% come back that are actually violative and a regulatory action is taken. A few more points. Violative residues may be restricted to an individual tissue, and this is difficult for producers to grasp. Why did I receive a violation notice on my cow, but yet I received a check for her? And that's because in some cases we only have the distribution of this residue in the liver or the kidney or maybe both organs, but it didn't make it to the muscle tissue, and therefore the muscle tissue is passed. In other, and this is kind of particular to a specific drug. If you're dealing with, for example, a sulfa drug, it's very likely that that drug is going to distribute to the muscle tissue, and you're going to have a carcass condemnation. With some other drugs, say penicillin, for example, that drug is unlikely to get to the muscle. There are a few cases where it does, but in general, it's limited to kidney or liver and kidney. So the carcass would be passed, but a violation would still be pursued, and the producer, the shipper, would still receive a violation notice. The other thing is that cattle with high violation risk conditions are screened even if they are condemned on post-mortem. So if you take your cow to the slaughter plant and they find tumor lesions of bovine leukosis in her and they condemn her, if she had any inflammatory lesions either or any conditions, antemortem conditions that would associate her with a potential residue on inspection, because she was presented as eligible for the edible channel, she will still be surveyed for residues, and if she's positive, a violation will be pursued. So you can still, even though your cow is condemned, you can still get notification on a violative residue. So to sort of tie this whole thing together, I think we all need to be involved in the One Health effort, and certainly antibiotic residues are part of that. We have lots of misinformation and misunderstanding about violative residues. We have many in the public, many in consumer organizations that believe that violative residues are associated with antimicrobial resistance directly in people, and that is certainly not the case. It's very unlikely that a human is going to get a significant level of an antibiotic compound to develop resistance in their gut. With that said, there are certainly some very important reasons why we need to avoid residues, and the concern with residues in food is not as much antimicrobial resistance as it is actually the direct toxic effects or allergenic effects on people of specific drugs. So we do have a responsibility from the public health standpoint to make sure that these compounds don't get into food. And we need to be actively engaged in that process. Certainly, from the standpoint of the reputation of our wholesome and outstanding dairy products, we want to do all that we can to maintain the confidence both in our domestic market and in our international markets that our products are residue minimized. So, Bob, I'll turn it back to you at this point. Thank you very much.
All right, Dr. Schultz, thank you very much for that informative presentation. I'll move right on. We're going to move to our next speaker. Our next speaker is Dr. Mike Apley from Kansas State University. He received his DVM and a PhD in physiology pharmacology from Kansas State. He's a diplomat of the American College of Veterinary Clinical Pharmacology. His practice background includes 2 years in general practice in central Kansas and 4 years in a feedlot consulting contract research-based practice in Greenlee, Colorado. Mike then joined the faculty at Iowa State University and was there for 9 years, and then from Iowa State moved back to Kansas State, where he is currently a professor in the Department of Veterinary Clinical Sciences. Dr. Apley works with veterinarians and producers throughout the United States concerning the use of drugs in food animals, and also in the area of beef cattle health with emphasis on feedlots. Now, Dr. Apley teaches in the beef production medicine course, the large animal medicine and pharmacology courses at Kansas State. His research interests include infectious disease, antibiotic efficacy and resistance, drug residues, and applications of drugs in food animals. Mike has also been the past president of the American Academy of Veterinary Consultants and the American College of Veterinary Clinical Pharmacology, and currently serves on the Veterinary Medicine Advisory Committee to the FDA Center for Veterinary Medicine. So Mike is very well positioned to talk to us about the pharmacology and use of antibiotics relative to some of the residues. Mike?
Thanks, Bob. Well, today I want to follow up Dr. Schultz's excellent presentation with one that's going to reemphasize a few points and have a little different view on it. And then at the end we can ask some questions. I put these slides in black and white because I think some of the issues are fairly black and white in how we address them. So I'm going to start out with a question here on extra-label penicillin use. And the question is that you purchase Procaine Pen G from the feed store and use it according to your typical dose of 5 cc's per 100 pounds under the skin. To avoid a residue, which of the following would be your best option? And number 1 is consult a label for the appropriate withdrawal time. 2, you'd stop right here because this is an illegal act. 3 is that it would be fine if you injected the drug intramuscularly instead because the subcutaneous route would be off-label. Or 4, I should only purchase this from a route truck because they will be able to help me with the proper procedure that others are using. So residues occur when detected concentrations of the marker residue are above the tolerance for the drug in that tissue, and that tolerance is based off a marker residue. And the target residue tissue is the tissue that holds the residue the longest, and this tissue indicates the status of other edible tissues in the animal. So it may be the liver, it may be the kidney, a lot of them is in the liver. So they may test the liver, and then if it's violative, assume that the rest of the carcass is also. So there can be legally acceptable concentrations of drugs in edible tissues, and except for a carcinogenic parent drug or its metabolites, which are also carcinogenic, if there is an established tolerance in tissues for that animal species. So for example, in dairy cows, if you have a residue detected of sulfadimethoxine, which is a sulfa that's okay to use in dairy cows if it's for a label purpose, and if that residue is below the tolerance, that cow is not violative. If there's a residue for gentamicin in the kidney, that's violative if any is detected because there's no approved use of that for that cow. So a drug without a label and a corresponding tolerance in the species, then any residue detected is violative. And one of the big points here is that as we get into mass spectrometers and some of the newer techniques that really can find these at very low levels, 0 is an ever decreasing value that we have to deal with. So what are the problems? In Bob Veal, primary residues found are neomycin and oxytetracycline, and many of those may be from using the neo-oxy combination product in milk replacer and not getting it withdrawn in an appropriate time. Gentamicin is one, flunixin, and tilmycosin, trade name Micatil, which is labeled for administration under the skin. It's a macrolide related to drugs such as Tylosin. So these are the primary ones coming out there. Now our big topic today is talking about residues in dairy cattle. And Dr. Schultz already addressed some of the ones that are an issue and how these are evaluated at the slaughter plant. I wanted to share with you some data, courtesy of Drs. Deborah Serra and Randy Arbaugh at the FDA Center for Veterinary Medicine, who have compiled these data. Here's flunixin, 21%, and as has been pointed out, it makes up 21% of these residues over 2005 to 2008, even though it is only evaluated within the animals that are sent on for further testing because the initial antibiotic tests at the plant are positive. The ceftiofur testing concentrations, tolerances changed, and also some testing techniques, and you can see in 2008 that resulted in more ceftiofur violations. Well, ceftiofur violations as they didn't have them before. It's very important that everyone's up to date and realizes that ceftiofur products, Naxel, XNL, and Exceed, have slaughter withdrawal times, whereas in the past when we had Naxel and XNL, we did not have a slaughter withdrawal time, but we do now, so it's very important to be aware of that. Penicillin remains our most common residue, almost 40% of the residues from 2005 to 2008. So let's pop back to that initial question I had. As I wrote the question, my intention was for you to pick that you'd stop right now if it was outside of a veterinary client-patient relationship, because that was an illegal act. We have to realize that penicillin can be bought over the counter, but if we use it at any other dose than what's on the label, which is 1 cc per 100 pounds, that 10-day label withdrawal time does not apply. So that's the reason for the veterinarian being involved in that extra label use, is that they then work with you to come up with that extra label withdrawal time. I also use subcutaneous administration in that question, and that's a flag because putting that drug under the skin rather than in the muscle can cause some significantly prolonged residue times, withdrawal times for that drug. So it's one that there's a reason it can get us into trouble, and obviously it does by being 40% of those violations. Sulfadimethoxine, trade name Albon, and then the generics is one that is approved for labeled use in dairy cows, and one of the sulfas you can use. Sulfamethazine does not have a dairy cow label, a lactating cow label, so using it in a lactating dairy cow is not legal even within a VCPR, and there's a few residues there. So what happened to cause these? The TRIMS database is a database maintained by the FDA that accumulates the results of FDA and state inspections of those that are involved or responsible for tissue residue violations. It's some really telling data that helps us understand what's going on. So these are the traditional causes of residues, and Dr. Schultz alluded to a few of these in his presentation of what can go wrong, but treatment records not maintained, not following withdrawal times, exceeding the approved dose without a veterinarian involved to establish that prolonged withdrawal time, extra label use by lay people outside of that VCPR, which could be related to those top ones causing a problem, and then ones that are fed or given by mistake. So why were these drugs used that caused these residues? About 88% were used for an illness. Preventative use was only 12%, so the vast challenge comes in that dairy hospital. Here's one that is pretty telling. Only 30% of those violative residues had a veterinarian involved in the decision process in working with that dairy and prescribing those drugs. 70% of the violations were because of use outside of a veterinary client-patient relationship. One of the trends I'm seeing in dairy practice as I talk with veterinarians is the increased use of written treatment protocols and more of a veterinary presence in the dairy hospital, actually overseeing and working more closely with crews as they treat these cows. And I think that's something that's very, very necessary to help us address this issue. So we have to extend withdrawal times in the case of extra label use. As Dr. Schultz pointed out, sometimes your veterinarian may consider it necessary to extend the withdrawal time even with label use when you have a very debilitated animal or one with some significant variations in how their body's working. But under absolutely no circumstances can we justify shortening the withdrawal period, only lengthening the withdrawal period. So here's some things that really get us in trouble. They include just blowing off withdrawal times, having no records, no protocols, no training of employees on protocols, and extra label use without the proper guidance. So under the Animal Medicinal Drug Use Clarification Act regulations, a veterinarian must be involved in any extra label drug use in the form of a veterinary client-patient relationship. Protocols are best developed with a working relationship between the veterinarian and the dairy person. This involves sitting down, evaluating the disease challenges on the dairy, how these diseases are diagnosed in the animals, and then coming up with treatment plans. My best protocols that have been developed with producers have been a partnership. And within that partnership, there has to be one very key agreement, and that is the only way a protocol is changed is if both people talk about it and it's agreed upon. Input's valid from everybody, but where we get in trouble is stepping off a protocol without the appropriate veterinary involvement. The records are important because we want to see what's happening as we put these protocols in place. It's not really a clinical trial, so we can't say the drug is actually doing this 100%. We're not 100% sure it's responsible for everything that's happening, but our records allow us to confirm it was used and what's happening on your dairy when we use this drug. Then if we go to changes, we at least have some idea of how things have been done, how they're going to be done now, and go onward. Where we run into problems in evaluating issues on dairies, both from residues and from treatment results, or even actual disease incidents, is when we don't have records and or we have protocols that are sometimes observed and sometimes aren't. So if you're going to go to the trouble of putting protocols in place, very, very important that everyone signs up and agrees that they're going to be followed. I mentioned earlier about veterinarians becoming more involved, even above what they are now, in dairy hospitals. And I think this is very important in having time for the veterinarian to work side by side with the herds people that are responsible for the actual placement of drugs in animals and the actual diagnosis. So what do I mean by extra label use? This is whenever a drug is used other than exactly as specified on the label. So this includes how much you give them, the route you administer it, how long you administer it, how often you administer it, the injection site volume, and then the treatment indication and the animal species and use class. One important thing in dairy cattle is we have some drugs that are banned for extra label use in cattle over 20 months of age, dairy cows over 20 months of age by the FDA, and that's phenylbutazone. And then we have some drugs that are banned for extra label use in lactating dairy cows, such as the sulfas. They can only be used for label use. In the FDA terminology, a lactating dairy cow is the same as a dairy cow 20 months of age or older. So if you have a dairy cow that happens to be dry, that is 4 years old, she's still considered a lactating dairy cow. It's still illegal to use a drug that's not legal in a lactating dairy cow. Some drugs today have some bans that are for all food animals, and this is why it's so important to have that veterinarian involved, because that extra label use can be a real slippery slope if we're not exactly sure what we're doing. So there's a clear hierarchy of uses in the AMDUCA regulations. They include use of a labeled product as labeled as the very first priority. We should always strive to do that whenever possible. And if that is a prescription labeled product, then a veterinarian has to be involved. If it is an over-the-counter labeled product used exactly according to label, then you may use that according to label directions. But the minute any of those things change, the veterinarian has to be involved before you can do that. Then there's the hierarchy, those next three steps. That last step, the fourth one, use of a compounded drug, is one that can get us into a tremendous amount of trouble. You've heard of generic drugs. A generic drug is an FDA-approved copy of a previously approved drug after the patent or marketing exclusivity period for that original approved drug is expired. A generic drug is manufactured, labeled, and marketed under FDA regulations. It has to meet the same regulations as the original proprietary drug. A compounded drug doesn't do any of that. So if you get a drug with a stick-on label and a plastic bottle with a cork in the top with some name made up for it, there are specific regulations that the veterinarian has to go through to make that legal. It involves a lot of things, including a very extended withdrawal time. All the components going into that compounded drug must be that they have to be labeled. So you can't use a bulk drug to put into food animals. It has to be compounded from stuff that's labeled. And again, only within a valid veterinary client-patient relationship, and only to products that are approved for use in either animals or humans. So DMSO is one that you're not supposed to find on dairies. Solvent grade in other food animals is not legal anywhere in a food animal. In some other animals, you could use one that is approved for animal use in an extra label manner, but DMSO is a drug where before you go putting it in any animal that's going to be sold for food, you have to be very, very careful. And of course, in dairy cows, they don't want to find that on your farm. So the vet's job in extra label drug use is to confirm it's legal, determine that the use makes sense medically, determine the extended withdrawal time, and the animals have to be correctly identified for observation of that extended withdrawal time. This is a place that things can break down, and you have to make sure they're very clearly followed. And then the vet has to provide the drug or the prescription. So some very clear requirements there of the veterinarian in those instances. So here's some practices I think just don't make sense. One is putting an entire dose of Pen-G in one spot. The long-acting penicillin you may hear about, I'm not a fan of it. Your veterinarian may have another opinion. I'm not a fan of it for the reason that the procaine portion is fairly rapidly absorbed, and the benzathine portion is what gives you the, quote, long-acting component of it. That benzathine portion is so low, the concentrations it achieves are so low, that in my opinion, essentially what you achieve is a long residue penicillin rather than long-acting. So I would encourage you to have a talk with your veterinarian about the different penicillin products, and I would propose that the long-acting penicillin on a dairy is something that would be a really low choice of mine. One example of a problem that I've run into is some people putting ceftiofurcrystalline-free acid in the neck instead of the ear or the base of the ear, and that is something that will result in some really, really extended withdrawal times, and it's an inappropriate administration technique going off-label for that drug. Another one is giving flunixin meglumine intramuscularly. I realize it's easiest when they're in a lockup to walk around the back, but that can cause some really significant extended withdrawal times when that drug is given off-label. And gentamicin, there's a reason why FARAD recommends an 18-month slaughter withdrawal time. That drug lights up some really prolonged kidney residues, even when given intramammary, and that 18 months isn't something that just is made up. And there's one established violative residue in the kidney of a cow where it was given 18 months earlier in a study. The compounded products, I don't mean to just dwell on this, but I wanted to come back and emphasize that point again. In the past, we've had compounded intramammary preparations, injectable preparations. I just don't see a place for those in modern dairy production because of the residue component and the fact that in some of those, if they're compounded from bulk drugs, they are illegal, and we just have no idea what's going on with them. Even combining two labeled approved products into one and administering them in an extra label manner requires some real careful consideration by the veterinarian as to exactly what's going to happen to efficacy of both of the drugs and also to the withdrawal time. And for those reasons, I recommend staying with the original forms whenever possible. These are just some excerpts that I've included from guidance 608.400 from the FDA on compounding. And the yellow part on this slide emphasizes that when compounding is intended to circumvent the drug approval process, that's where they really get an issue with that. And there's a huge difference between a generic drug, again, and a compounded drug. Compounded drugs are not produced under good manufacturing procedures that are inspected by the FDA. The guidance states that pharmacies and veterinarians who engage in activities analogous to manufacturing and distributing the drugs for use in animals may be held to the same provisions of the ACUS manufacturers. That means you're going to be held to a really, really high standard. This one just says the FDA has been affirmed to have the authority to regulate compounded drugs. And this one just points out that you cannot compound from anything other than an approved drug. So the takeaway from my part on the compounding would be that it's very complex, and I think compounded products are absolutely a last-ditch effort. So in conclusion, I think it all comes down to developing a treatment protocol. This involves the definition of what you're going to treat, the regimen developed with a qualified veterinarian, the outcome definitions, meaning how am I going to decide this animal is a success or a failure, and then the animal disposition. What are we going to do with her if she's a success? How do we maintain the withdrawal period? And then what do we do with her if she fails the treatment? And animal ID is so incredibly important. Adherence to protocols, and then monitoring adherence and outcomes through records, and be sure to double check those records before selling an animal. So with that, thanks for the opportunity to talk with you today. And Bob, I think we're ready for questions.
All right. Thank you, Mike. I see one question there. I guess either Mike or Craig, when you talk about records, does this refer to written records and not some kind of eraser board type records, disappearing information? And if that's the case, how long do farmers need to retain written records?
Go ahead, Dr. Schultz.
Well, I have not ever seen any regulatory requirement on the FDA's site requiring a specific amount of time. I guess it boils down to the turnaround time on an FDA investigation, which is often up to a year. With animal ID requirements right now, if the pending proposed final rule for animal ID goes through, the record-keeping requirement for animal ID will be 5 years. So I think less or more is better than less. You certainly want to have records around long enough that you can provide them, and that they're in an organized fashion, so that you can actually refer back to these and have something that's in like chronological order that you can look at and use those. Because I think the records are the producer's best defense with an FDA investigation, even in the event you were subjected to one. If you have adequate records that demonstrated that your intent was to use these drugs properly and to follow protocol, it's certainly much easier to deal with an investigator than when you basically have nothing at all to offer. So, Mike, do you have anything to add?
I think your comments on the fact that it's your best defense is absolutely on the money, and having both the protocols and the training. It's one thing to have a violation when you've had things in place and there was a slip-up versus just not having anything. And I was just trying to scroll through AMDUCA real quick here to see if I'm remembering there was some type of perhaps requirement. I think you're right on time for extra label use, but I think they definitely need to be held. I'd be comfortable keeping them for at least a year or so. And
now, would that, Mike, be a year after the animal has left the farm, or a year after the animal has been treated?
Well, in the dairy, basically your chance while they're on the farm, your only chance would be, for milk. And then once they leave, I'd say a year after they leave. I just pulled up Amduca, and the veterinarian has a record-keeping requirement with what has to be on the record for the extra label use. And under Amduca regulations, they require records be kept for 2 years, whereas otherwise required by federal or state law, whichever is greater. So the veterinarian has an obligation to keep them for 2 years after prescribing extra label use. The chalkboard comment is really appropriate, and there are some really nice computer treatment records used on the feedlot side. They may not be economically viable for some, but they make it easy to keep those records for a long, long time. I would, a couple years anyway, to keep those to show what happened and to go back and clarify things. And hopefully these records are the source of your veterinarian's visits too, as you look back on what's been done.
So something as simple as just keeping a notebook and having pages and adding pages to a notebook is probably adequate.
Yeah, what you need to do is design it so that you can go back and check those cows very easily. It should be by cow.
Mm-hmm.
Where you can go back
do either of you know of like a website, an extension site that might have treatment records? If you happen to come up with, you could type it in.
Not right offhand.
I don't either. I
know
to go dig.
The University of Minnesota has a program about drug use, and this is dairy-based. I don't know if it's something similar to what you're talking about with the feedlots, Mike, but I'd have to go back. Dr. Rapnicki, Paul Rapnicki up in University of Minnesota had something along that line. We have another question. Can you feed colostrum from cows that were dry treated, according to the label, I'm assuming a commercial product, to Bob veal calves?
Mike, do you want to start?
Milk, fluid milk from cows is not, and I've just had this conversation with the FDA, as I interpret this, milk replacer would be considered a feed. Drugs that go into the feed are classified as feed additives, or that go into milk replacer. A drug that goes into milk replacers is considered a feed additive, so milk replacer is considered a feed. Whole milk is not considered a feed. So if the question is it an illegal extra label use in a feed to those calves, as I understand it, no. With that, I would say that I would be very clear as to what drugs are in there and work with your veterinarian, and they would probably work with Food Animal Residue and Depletion Service, formerly Food Animal Residue Avoidance Data Bank, FARAD, and determine what those low doses might cause in residues. But the issue of, wow, what kind of residues can we cause with that are very, very important, and also the realization that even if you're flash pasteurizing that before feeding it to those veal calves, many of those drugs will survive that intact. Dr. Schultz, you have?
Yeah, I would add that surprisingly, we still see, at least up until 2008 when I was in the plants, we were still seeing streptomycin residues in calves and cows that were apparently coming from dry cow products. And there aren't very many products left with streptomycin in them. I think the Tomorrow product is about the only one. But that for whatever reason, we would find Bob veal calves with streptomycin residues, and I could only conclude that it came from milk.
And that would be an example of one with no clearance in those calves, so if they detect it and identify it, it's violated.
Yeah.
So, it would be with great concern for being able to control the residues on those calves that I would do that, and realizing full well the value of that milk and what milk replacement costs.
Yeah.
Okay. This is just a comment that says DHI electronic records allows entry of treatment records, so that might be an opportunity for people to use.
And Bob, I was going to mention, if I've got a sample treatment record deal that we use for smaller beef producers that goes by cow, and basically you can have a page per cow and or animal, and go down through and enter dates, and basically we have temperature, condition, drug used, and then you could flip back through and find her number to check and see if there's anything there. But the ability to do a post-shipment check is incredibly important on those cows and can help avoid a lot of issues. And my email is just mapley, M-A-P-L-E-Y, at ksu.edu, and I'd be happy to send that to anyone who'd like to.
All right. Thank you.
We have some dairy practice, some of our larger dairy practices here in the Northeast. Well, Pennsylvania and New York I have most of my experience with, but they have some pretty well-developed treatment records that are a part of their treatment protocol handout to their producer, and they're to some extent farm specific, but they're sort of based on a template, and then they can add farm specific data as necessary in the treatment protocol. So they're very functional and work very well, and we've had some good experiences with them on residue investigation follow-ups.
And again, Craig, you made the comment that one of the biggest challenges with the residue follow-ups is that the origin farm generally has no records.
Right.
So records is really probably one of the biggest take-home messages from today.
I think that, Bob, and that if you're a dairy producer and your veterinarian is not intimately involved in your hospital system, they need to be.
Agreed.
One of our other challenges as we wrap up here, in many areas here in the Northeast, there are regions that have very low veterinary service, and so there's many like small beef operations and smaller dairies that are somewhat challenged in having routine veterinary service, and there's a lot of reasons for that. But this whole residue issue and antibiotic use, and certainly the larger picture as this comes up to antibiotic resistance issues, I think this is a really important issue for us to grasp and get these owners and producers connected with veterinarians in some way to oversee this.
I think even if some banded together to have the opportunity to bring some people through would be a really good initiative to take. The involvement of a veterinarian in drug use today, I think, is just of paramount importance.
All right. Well, our hour's up. Thank you very much.